All right, so welcome to the History Center of Lake Forest, Lake Bluff's presentation on the history of vaccines. And I particularly want to thank Tom Thomson for helping us put this together. He's been incredibly resourceful. And we've got a couple of slide presentations that we did at the beginning of the pandemic around the history of pandemics and some other interesting sort of scientific historical narratives that we'll link to at the end of the presentation as well. And I know, Tom, you had a couple things that you would like to say before we kick off, so.
Sure. Well, good evening, everybody. And Carol, thank you very much. I'd like to thank you for hosting tonight, and also thank the History Center for the invitation to put this program on. Alex Schneider, who's in the background there somewhere, was very helpful in assembling the slide set. And then certainly David Forlow, our president of the History Center, has done a wonderful job assembling a board and assembling the great crew that mans the History Center. If you haven't been there, you should visit. There's much to learn, see, and appreciate. I think that my first vaccine exposure as a child was in the 1950s when I received the polio vaccine. I distinctly remember not only being surprised, but frightened when a stainless steel tray with 3 large glass syringes and a needle that seemed like it was 5 inches long on each one of them came into the room with a nurse. And it was about 10 minutes later, after they had to call the pediatrician in, probably the general practitioner at that time, in to catch me so that they could give me my 3 polio vaccinations. From an epidemic standpoint, I think my first recollection was in the 1960s. During the Hong Kong flu. It happened to be the winter that my sister was married, and she spent her honeymoon with bedridden influenza. The true motivation for my interest in microbiology and vaccines and epidemics was really solidified in the mid-60s, mid-70s, 1976. That was the year that we had the swine flu pseudo-epidemic. There were 2 soldiers in Fort Dix, New Jersey that came down with swine flu. They thought it was the exact same influenza strain that caused the 1918 pandemic. The United States mobilized the vaccine effort. President Ford at the time promoted the vaccine by having his whole family vaccinated on television. About 65 or 70 million people were vaccinated. We did not have an epidemic that winter. That's why I refer to it as a pseudo-epidemic. Coincidentally, that same year, the Centers for Disease Control was called to Africa, 1976, and they used electron microscopy to identify the Ebola virus in patients. This was the first documented Ebola virus in Africa. During that investigation, the Centers for Disease Control bled about 300 people. So they collected serum to look for antibodies to Ebola. As the CDC does, they put everything in a freezer and save it forever. Half a dozen to 10 years later, when they were investigating the outbreak of HIV, they went back to this refrigerator, this freezer, and pulled out these sera, which were obtained almost a decade before the HIV epidemic. And they tested them for antibodies to HIV, and the majority of them were positive. So during the pseudo-influenza outbreak, detecting Ebola, there was another overlapping epidemic that was still 5 to 10 years away from being discovered. So that is a background, Carol. Why don't we proceed in tonight's program?
All right, that sounds great. All right, so the goal is to have this be a conversation. You should, on your screen, be able to click on the Q&A button at the bottom and ask questions during the presentation. We also will have a Q&A section at the end for anything if you would rather have it be more conversational and ask. But what we want to do is sort of give you an overview. Obviously, every one of these slides could be an hour-long presentation. So this is going to be very sort of top level in terms of what we're going to discuss. And so where we begin is, okay, so how long have humans been trying to immunize?
Well, as far as we can tell, immunization, as you know, is taking an organism, which is a pathogen, or part of an organism, and scratching or inoculating or adding that in one way or another to an individual And by doing that, you use immunity or protection from the disease without actually causing the disease itself. And although the pox virus probably originated multi-thousands of years ago in Africa, it was in China and India 1,000 to 2,000 years ago that the process of variolation began. That's where they took pus, purulent material, infectious material from a skin lesion of somebody who had smallpox and would scratch that into the skin of somebody who did not have smallpox. And that small amount of material, in many cases, was too small to cause fatal disease, but large enough to cause mild disease that induced immunity. So this variolation is our first evidence of immunization. Now, in addition to scratching it into one's arm, they would also dry the material from these pustules and then blow it into the nose of people. So this crusty, dry material would be blown into the respiratory tract, and that too induced immunity and caused disease far less frequently than if you were exposed to a person who actually had smallpox. The Latin term for pustule is variola, which is the origin of the term variolation.
All right. So as long as we're discussing this linguistically, what is the difference between a vaccination and an immunization?
So thousands of years went by before Edward Jenner, who himself was variolated to protect him against smallpox. Edward Jenner noticed that dairy maids who milked cows that had cowpox would develop a lesion on their hand, but they never developed systemic, serious, fatal disease from human smallpox. So he did a trial with one boy where he took material from the cowpox, scratched it into his arm, waited a few weeks, and then exposed him to human smallpox. Not a trial that I think the FDA would allow us to do today, but nevertheless, it was a trial that was done. When the boy was exposed to smallpox, he never acquired the infection. So this was the beginning of a new type of immunization, and VACA comes from the word cow, and so Jenner preferred to call that term vaccination. So vaccination has become the way we refer to the delivery of a vaccine or of an immunizing material into a person. Now, this may be into their skin, into their bloodstream, into their respiratory tract, into their muscle. There are many different ways it can be delivered, but vaccination is the process of delivering this material into the patient. Immunization is actually very similar, but it includes vaccination and the person's response to the vaccine developing immunity. So immunization is a broader term. Vaccination simply refers to delivering the material into the patient.
Excellent.
Interestingly, the smallpox vaccine has been just about the most successful vaccine in history since in 1980, the World Health Organization declared smallpox eradicated from Earth. And it's all because of the vaccine.
All right. What are some of the other early vaccines?
Well, there were many early vaccines that were successful, and we'll talk about those in a minute, but the vaccine trial that gets the most exposure is that Pasteur conducted. Pasteur and Koch and other early microbiologists developed methods for isolating bacteria or allowing bacteria to grow on artificial media. So instead of not knowing what the infectious agent is, or possibly seeing the infectious agent only under a microscope, Pasteur was able to grow many of the infectious agents on artificial media. And one of the infectious agents was anthrax. It's a bacterium that goes by the scientific name of Bacillus anthracis. It's primarily a disease of animals, but a devastating disease because it was passed from animals to animals and it would kill your entire herd. Pasteur took the Bacillus anthracis in the laboratory and passed it from medium to medium to medium until he was able to change the organism enough that it was no longer pathogenic. He then took that non-pathogenic form of Bacillus anthracis and he conducted a trial. He took, according to lore, he took 25 animals and injected them with this vaccine, and he took 25 matched animals and did not inject them. He waited 30 days and then challenged all of them with Bacillus anthracis. Those who were vaccinated survived. Those that were not vaccinated did not survive. Really, in my mind, the first randomized, placebo-controlled study that we have evidence of being conducted.
All right, so you talked about this a little bit, but I mean, clearly, we start small. We always start with the smallpox and we get a couple other diseases under control in the next decades, and then it sort of explodes. So walk us through a little bit of that model and why it accelerates at the rate it does.
Yeah, it really did expose. The first diseases that were pinpointed for vaccine development were those bacterial diseases, primarily bacterial diseases. The smallpox is a virus, but these other early ones were bacteria. And these organisms were grown on artificial media, so we had a vehicle for vaccine production. These were also infections that were primarily adult infections. So these were large pandemics that scourged the earth multiple times every decade, and they did this not just for centuries, but for millennia. So when we talk about typhoid and cholera, in addition to smallpox, these were early targets for vaccination. And so that was the first sort of phase of vaccine. The second phase were really common childhood vaccines that killed many children, before they reached adulthood. And this would be pertussis or diphtheria or tetanus. And then later, when we determined how to grow viruses in the laboratory, we actually then had the virus, the same way we had a bacteria on a culture plate. We now had a virus later in the 1900s, and they were able to inactivate those viruses and make vaccines against the common viruses. So then we had childhood diseases early on. We had childhood diseases later on with viruses. And then in the 21st century or the 2000s, we had genetic techniques that allowed vaccine production for microorganisms that vaccines had not worked for before, or in unique populations of people where vaccines did not elicit antibody production. So we now had stronger vaccines that were safer, that were developed against things like shingles and pneumonia and human papillomavirus, the cause of cervical cancer. All of these are modern vaccines, and we'll talk about their development in a minute, that were the final phase of vaccine development, or the current phase of vaccine development.
All right, so there's a lot of anti-vaxxer narrative that is floating around right now. There's a lot of sort of naysaying around whether the effectiveness of them and whether or not they introduce as much problems as they solve for. Let's talk a little bit about why do we need vaccines?
I hope there's some young budding scientists or young budding historians that are listening tonight. As a scientist, I'm pleased and surprised that when I go back and I read history, it's much like interpreting scientific data. Data is data, and you look at it and you conclude scientifically what the data is trying to tell you. Does this vaccine work, or does this vaccine not work? When you read history, especially the farther back you go, you get historical data, and the data can be conflicting. Some people saw the data one way, some people recorded the data the other way, and as a historian, you're somewhat like a scientist. You have to take the data and make that interpretation. So what I'm really doing tonight is, as a scientist and as a pseudo historian here, I am reviewing what I find of past information about vaccines, and I am giving you the data, but primarily I'm interpreting that data for you. We don't have time to go through all of the data. So, live with my interpretation. Doesn't mean that you can't interpret it differently, but remember, data is data. And you can't lie about the data. It's what it is. But shockingly, the life expectancy of those of us in the United States, even 100 years ago, was drastically shorter than it is today. I remember my mom, who had her 90th, she was born in 1921. And when she had her 90th birthday, I looked up in the US life expectancy tables. And when she was born in 1921, her life expectancy was 49 years. You know, she eventually lived to be 95, but she did have three other siblings, so 4 in their family, 2 died in childhood because of childhood diseases. So if you looked at the average life expectancy in her family, her sister is still alive in her 90s. It's probably about 49 years. It's probably about 49 years. So as you can see by the data that's on this slide, one of the main reasons that life expectancy at 1900 and after we took care of poxvirus and cholera and the plague, we had ways of controlling those. Most of the life expectancy was controlled by the death of children. You know, to live to 21 was a lot more difficult then as it is today. So you can see here that in 1900, 30% of all US deaths each year were in children less than 5 years old. And if you look in 2000, it was only 1.4% of all deaths each year are a result of kids dying before they reach 5 years old. And much of this progress has been made because of the vaccines that we discussed briefly in a slide before. In addition, there are some stark examples of vaccine efficacy. 1918 to 1920, we didn't even know that influenza was caused by a virus. And upwards of 50 million people possibly died worldwide. In 1952, there was a large polio epidemic. This is before the vaccine. And 3,000 patients in the United States died of polio. Now, both of these diseases are either wholly or partially, but most partially, controlled now by vaccination. Polio, we're on the road to eradicating polio from the earth. It's been a difficult battle over the last 20 years, but eventually health experts think we will get there. And influenza has been controlled if you take a vaccination, and also if you do get sick, we have antiviral compounds that can be used to treat influenza. So vaccines have had a huge impact on our life.
All right. This is just a brief chart that we've got that just shows some of the numbers around number of cases and the deaths. Anything that you want to pull forward from this chart? Just a couple of samples.
Yes, so this is a representation. I think that was 2009 of pre and post vaccination morbidity, mortality, and there was cost data in this article also. So morbidity is disease, mortality is death, and cost is obviously dollars. And you can see that the average number of cases and deaths before 1980, in the early years of in the 1900s, were significantly greater than the number of diseases and the deaths after 1980. Now, this is average per year. One number that stands out in my mind here is the number of fatalities due to pneumococcus. This is pneumonia, and we have the pneumonia vaccine. And there are 2 observations here that I make. The first is that there are way more than 5,000 cases every year. It's not a reportable disease if it doesn't cause meningitis. And most of the fatal disease is caused by pneumonia that disseminates to the bloodstream, and people die of sepsis. So that number is artificially low. The number of deaths is probably fairly accurate, and the reason that it is high compared to the deaths in pre-vaccination years is that not everybody gets their pneumonia vaccine. the number of people that get that vaccine, at least in the elderly generations, is less than that get the influenza vaccine. So the pneumococcus is somewhat of an outlier, but it still leaves room for improvement if we're able to apply that vaccine more completely in the needed populations. Additionally, cost is not on here, but this article talked about cost, and it is felt now that vaccinations each year prevent 20 million diseases collectively through all the things that we vaccinate for, and 42,000 deaths. And this amounts to $13 billion of direct costs per year, and significantly much more societal costs if you throw in the indirect cost savings. So vaccines, once again, have had a tremendous impact, on our life expectancy, our health, and even our finances.
All right, so we have a brief film that I'd like to run around a historical moment and why safety regulations are so important. So Tom, is there any setup you want to do before I run this?
No, go ahead. I can make a comment afterwards.
All right.
Hi, I'm Tom Carney Jr.
And I'm Janet Carney O'Brien.
And we've been invited to spend a little bit of time talking about our father as a pharmaceutical experience.
Well, Tom, Janet, thank you both so much for taking the time to share a little bit about Dr. Thomas Carney, about your father. From what I know about him, from what you've already shared, he was a very extraordinary individual, and I'm really looking forward to learning a little bit more about him. So let's go ahead and dive right in. I know that your dad was involved with a lot of medical advances in the pharmaceutical industry from the 1940s to the 1970s. He had a very long, rich career. What would you say are some of the most important or recognizable projects that he was involved with?
Well, I could start with the one that I know is the first recognizable, at least for a long time in the day, was Cervicaine, and he developed that application, that product, when he was a fellow at University of Wisconsin. And it's a topical anesthetic that was actually a result of a Surgeon General plea during World War II. This is in 1942 for a topical anesthetic for burned soldiers coming back from the war. His lab developed that, and it was part of his most rewarding, one of his rewards for going into pharmaceuticals, and that pretty much put him on the path of medical research. And then from there, he went to Eli Lilly. I think Tom has sort of a rundown of the things that came out of Lilly.
Of course, we're talking 50 years ago, so the brand names have been pretty much disappeared since then. They've been supplemented by advances on what he did. Probably one of the most long-lasting ones is the biosynthetic penicillin, which was developed in the Lilly labs under him, that allowed the mass production of penicillin. He was involved in the early non-drowsy antihistamines, clopyranol and histidil, analgesic, Starvon, methadone, lots of cancer research. He also got Lilly involved in agriculture, by the way. Thought very pragmatically that since they were testing all these products on animals, why not use the results to see what they could do for veterinary medicine? So they developed the Greenfield products under him too. But probably the most dramatic and long-lasting was Lilly's involvement in the Salk vaccine that ran across the fact that the NIH at the time was the only place that had a lab for tissue cultures, tissue development. And he thought that would be a good thing for a pharmaceutical lab to have too. He didn't have any particular use for it, but he thought it would be worthwhile. And a couple of years later, when Salk vaccine came along requiring the mass production of polio virus, Lilly had a ready-made tissue culture lab. And so they ended up being able to develop a leadership role in the Salk vaccine. At one point, we're producing about 60% of the Salk vaccine in the country.
Mm-hmm. That was back about 1955.
55, well, yeah, 55. 54, Salk was announced. 55 was when it was licensed.
Yeah, so Lilly became pretty much the premier developer and commercialization of the Salk vaccine, which really went out to national distribution pretty quickly because they were already set for it.
Sure, yeah, and that's you know, so important to have that. It's really interesting that they had that huge percentage right there that they were, you know, able to take on that challenge during that time period. And of course, for many people right now who are experiencing the current COVID situation, they also lived through the polio outbreak, and you know, we're keeping tabs on the Salk vaccine. So thinking back to that time period, do you personally remember anything about what it was like to be living during the polio epidemic?
Janet probably no, since she was 0 at the time. Yeah. Janet is much younger than I. I was born in '45, so polio was a summer, a periodic summer disease. It didn't occur, surged in the summer, but not every summer. And I think '52 was the last real one before the Salk vaccine, and I would have been 7 there. So I don't have a whole lot of memory of you know, worldwide, community-wide. I do remember that I developed scarlet fever in the summer of '72, and because it was also the polio time, I ended up spending the summer in bed. And in 1952, we didn't have a television, so that wasn't fun for a 7-year-old. So I have that memory, but not specifically of polio. The more dramatic one was in 1955. The Salk vaccine was licensed for distribution in early April of 1955. And Dad had been director of research, he'd been director of quality control, he'd been director of production, and he was about to become vice president of research development and control at Lilly. And so he was going to be in charge of the whole rollout of the Lilly vaccine. And it was about a week after the announcement of it's been licensed. We were driving in a car someplace in Indianapolis. Dad says he was driving my other sister Sheila to a music lesson, but Sheila would have been 3 at the time. So we've all agreed that's probably not true. But the radio news was on, and there came this announcement all of a sudden that there had been reports of numerous children who had been injected with the Salk vaccine coming down with polio. And of course, Salk was a live vaccine, started live vaccine, which was killed, and so it was dead vaccine that was injected. But obviously there was a risk. There were protocols in place, but the announcement was made that the Salk vaccine was causing polio. Well, Dad was personally invested in this, obviously. Lilly was one of 5 or 6 labs producing it. So he didn't say a word, but made a quick U-turn on, he describes it a one lane street, and I don't remember that, but I do remember the U-turn and the absolute silence going back to the house, dropping me off without a word, and then heading off to the lab to find out what was going on. The announcement didn't say where the vaccine came from, which manufacturer it was, just that the Salk vaccine was infecting kids. And it turned out that there were thousands of kids who got sick, several hundred got paralyzed, and I think the number was 10 who actually died as a result.
Yeah.
And obviously you can imagine that this news, even without social media, would have had a dramatic impact on the Salk vaccine. Mm-hmm. Clearly a business impact on Lilly, but a profound personal impact on Dad, since it was, in Lilly at any rate, his baby. He was responsible for that. And we didn't see him for a couple of days until they finally figured out generally what happened. And Janet?
Came to be known as the Cutter incident.
Yeah, it turned out that the one lab was the Cutter lab, a small lab in California.
Yeah. And they had followed.
Remarkably, they had followed the government protocols, but Lilly had, under Dad, higher protocols.
Well, Lilly followed the Salk protocol. I mean, Salk had his own very stringent protocol, and Lilly followed that, but it wasn't necessarily required of all the other smaller labs that started picking up on this whole vaccine rollout, and that they were able to isolate it at least to one lab in California, and that's what all the work was involved in. And then they ended up suspending the vaccine effort for several months while they went back to try to figure out what to do now. And that's when it really was kind of a turning point for vaccine development and federal regulation. The feds came in, FDA actually said, okay, we really do need federal guidelines in vaccine development. They came in, put together guidelines, which people accepted, and it actually, after several months, they started the vaccine rollout again. I mean, they were able to regain public trust, which was pretty remarkable. I think that with all the guidelines came in very well communicated, and people went back to trusting that, okay, we trust it's okay now. And
that's very impressive.
So, yeah, we eradicated polio because of that.
Yeah, that is very impressive that they were able to turn around and regain that public trust. It's a very delicate thing.
Yeah.
So a lot of really interesting results out of the Cutter incident. So just to kind of wrap up, you very generously shared some of the segments of your father's memoirs with us. What would you say really motivated him to create a record of his life and his career?
I think there were a lot of things. It's difficult to say complicated, but there are a lot of things involved in that. He fully recognized that he was in the center of what he described the golden age of pharmaceutical research and development. That Lilly was one of, if not the largest pharmaceutical house in the country at the time. He started as a bench chemist and ended up as vice president of research and control, and he had the opportunity, and he saw it as a blessing and an honor, an opportunity to encounter a whole range of truly great people in that area. Nobel Prize winners. We had Nobel Prize winners dinners at our house, and it didn't mean anything to us because we didn't know what a Nobel Prize was at the time. But it was that personal, visceral, hands-on experience he had with the truly miraculous development of pharmaceuticals at the time. And he spoke of one particular individual, Dr. George Close, who was director of research at Lilly when Dad went there and was involved in moving Lilly to the forefront of insulin production. He was also a pioneer in cancer research, and they were very close. And he talked a number of times about how he talked with Dr. Close about writing down his experiences so this experience would not be lost. These were giants, and he recognized he was standing on their shoulders, and he wanted people to write these things down. Well, Close didn't, and he died with all his memories to himself. And I think Dad, his view was, at least for the family, and he never published. And if you read the whole thing, he was pretty honest about his views of some people and some things. And I think he probably didn't want all of it published, but he wanted at least his family to know what went on and the excitement he felt about the times he was in, and that he was honored to participate in. And I think he truly saw it as that way, as an opportunity, not something he did, but something he took advantage of.
Yeah, and I can add to that too, that as Tom was saying, he saw his place in history or medical history as part of a continuum, and he benefited from the knowledge that he gained from people before him, and he wants to pass his knowledge on to the people beyond. But he did end it, I thought it was kind of striking that his memoirs ended on a note of gratitude, that he was just thankful that he was born just at the right place, the right time, right opportunities. And he was grateful for that. So I saw not only this as sort of he wanted to make sure that the history was down, but he also wanted it to be like a personal thank you letter. It read to me a little bit like at the end, like a thank you letter that he had this life. So it was a little bit of both, that he basically shared it with the family, and that's now in writing, so we have it.
A special for you both, for your siblings, for the rest of the family. That's really incredible. Well, Tom, Janet, thank you both so much for taking the time to share a bit about your dad and his work and his legacy. Really appreciate it.
Thank you.
Thank you for the opportunity.
Well, and Tom and Janet, thank you for the wonderful recollection. I think what your story highlights is one of the many incidents that has matured the FDA over the years. Really, as a modern regulatory agency, it's got its start around 1906, following the book The Jungle by Upton Sinclair, and they came up with regulations applying to meat sent across state lines. And since then, the essential oils that were supposed to cure cancer and drug content, purity, and safety introduction of prescription drugs that you could only buy with a prescription, the Cutter incident that you talked about, the thalidomide issues in the early 1960s. There have been many steps that the FDA has taken based on experience to make it the organization that it is today, which very thoroughly vets all of our new vaccines through trials that are overseen by the National Institutes of Health. One last thought about polio. It's really an interesting virus. It emerged from the Coxsackie group of viruses that some of you may be familiar with, because kids still do get Coxsackie virus infections, hand, foot, and mouth disease, for example, during the summer. And it's been around for millennia, but it really emerged as a serious epidemic and organism in the early 1900s. And what happened was that before then, hygiene and sewage treatment was so poor that everybody was exposed to the polio viruses within the first months of their life. Sewage, human sewage was thrown out into the sidewalk and into the streets, and everybody was exposed to this every day, even newborn children who still had antibodies in their blood from their mother. The passive immunity that they acquired that lasts, 6 months to a year, protected them against polio disease. So when they came in contact with the polio virus in the streets, they were actually immunized. And that immunization, natural immunization, lasted their whole life. So there was not a lot of outbreaks of polio in older children or adults until treatment of sewage became sophisticated and nobody was exposed to the polio virus early in life. So all of these outbreaks that we recognize are really within the last 100 years. And they were serious enough that, based on lessons we learned from developing an influenza virus during the Second World War, Salk and Sabin subsequently developed vaccines for the polio virus. Pretty interesting story. The slide and the question that you have here continues to stress how important vaccines are. And this just takes the big picture of vaccination and compares that or adds that to the effects of antibiotics, sanitation, surgical techniques, anesthesia, all these things that have amounted to significant progress in the last 100 years for human health and longevity.
All right. So, one of the things that got me interested in this about 40 years ago was I was in the Peace Corps, and part of my job was to actually go around and immunize with the World Health Organization in some of the most rural communities in the Central African Republic. And so one of the things I know helped with that ability to actually get out there and do that was the ability for us to manufacture vaccines that didn't require massive refrigeration, that were shelf stable enough that we could get them out into rural areas. So Tom, talk a little about what that process is like. There are different ways to manufacture vaccines, and sometimes it's an evolution and sometimes it's just a choice based on effectiveness.
Yeah, so the production of vaccines, as we all know, has changed for the better significantly over the last 100 or 2 years. The early vaccines were whole organisms that were made non-pathogenic or were killed, and they were used to immunize individuals against that particular disease. And certainly the original smallpox vaccine with variolation and then vaccination, and polio, an inactivated whole virus vaccine, are examples of these early vaccine types. Now, they gave excellent antibody production, but the vaccine production was slow because you had to prepare the whole virus, and the side effects were more significant because there were more particles that you were injecting into somebody's arm. Following this, the whole organism vaccines, they began to realize that it was just part of the organism that was most important in developing immunity. And so they began isolating that portion of the organism. So that would be called a partial or a subunit vaccine. So for example, with influenza, we know that one of the spikes on the side of it, which is called a hemagglutinin, is important in the attachment and infection of human cells with the influenza virus. So when we make the influenza vaccine, we just make hemagglutinin for that vaccine. We don't include the whole virus in the vaccine. So once again, this production is relatively slow. You get pretty good antibody production, but there are far fewer side effects. Now in the last 20, 30 years, we've been able to genetically engineer our vaccines. So we take the gene from the organism that is responsible for producing the immunizing chemical, and we either put that gene into a human and they use that gene to make the protein, the immunizing chemical, or we take that gene and we put it into a bacteria like E. coli or a yeast like Baker's yeast. And that gene will be, the protein from that gene will be produced during the growth of the E. coli or the growth of the Baker's yeast in very, very high quantities. They then isolate that protein that they're going to use as a vaccine, and they use that to inject into an individual. So this makes it much more quick, much more rapid to develop the vaccine. It gives above average antibody production, and it has fewer side effects as the subunit or partial vaccine do. So we've made significant strides in both the side effects, the efficacy, and the improved turnaround time and safety for these vaccines over the decades.
All right, so I know that when we did our presentation last October on sort of the history of pandemics, that one of the things that we were specifically talking, and actually I guess it was even longer back than that, because we were really focusing on HIV and AIDS, that one of the things that had really changed under the process of developing the treatment protocols for HIV was the speed at which we were able to do it. Although it felt very slow for the population affected, it was in fact faster than we've ever turned something around. And since then, I know that it's accelerated. So can you, I know that this is one of your areas of specialty, talking about how we were able to get so rapid in terms of our response, so that within 9 months, we were able to get a vaccine for COVID.
Yes, so as illustrated in that previous slide, we can produce the vaccine very, very quickly. And we have molecular methods now for surveying an organism that's a pathogen, figuring out what part of that pathogen is important to address with a vaccine, and then constructing the vaccine from that molecular model. And then once we have that construct, we can make the vaccine literally in weeks or months through our molecular methods. We still, however, have the extremely important steps of determining the safety and the effectiveness of this vaccine. And according to the FDA, that goes through 4 phases. The first phase is where a few volunteers are given the vaccine to determine a dose and also to make sure it's safe. There have been animal studies done to make sure it's safe up to then. This is the first human trials to make sure it's safe, and that's phase 1. Phase 2 is where they take a larger number of volunteers and they give them the vaccine, and they get exposed through their natural activities to the infection, and they address whether or not this vaccine prevents that infection. So that would be phase 2 efficacy, and they still monitor safety certainly during that. Phase 3 is where they do a large controlled trial. So it's usually hundreds, and in the case of COVID, it was tens of thousands of patients were split into 2 groups, those that got the vaccine and those that were given placebo. And nobody, including the patient, the doctors, nobody knew which group were placebo and which groups were vaccinated. They then let them go about their normal activities, and they did this in various parts of the world where the COVID virus, the virus that causes COVID, was circulating. And then they tracked them and tested them periodically to see who was protected and who was not protected. So that's a phase 3 trial. They then take all of this information and they put it together into a new drug approval form and submit it to the FDA that reviews it and then says yes or no. That process, phase 1 through the NDA, is lengthy because of the human trials that are absolutely necessary to document the effectiveness and the safety of these vaccines. They have to produce the vaccine, they have to distribute the vaccine, they have to administer it, and then people have to develop immunity. And as Carol referred, this was a years, if not decades, procedure in the past, but it has been significantly improved by our molecular methods and our ability to streamline the FDA process as much as possible, under pandemic conditions. And then there's phase 4, where for years afterwards, people given the vaccine are tracked, and safety and outcome has continued to be monitored to make sure that there aren't minor side effects that did not show up when you gave it to 10,000 people that might show up when you give it to 10 million people. So that's the important and somewhat time-consuming pathway to a new drug approval.
All right. So one of the things that I know everybody is hearing a lot about right now is that there are variations now on the COVID-19 virus. And so those mutations, let's talk about why they happen, and let's talk about how the FDA and manufacturers navigate them in the process of developing vaccinations and immunizations.
Yeah, this is so interesting and so important, but it does require a little bit of understanding of the virus itself. All of you have seen pictures of the coronavirus. It has these spikes that come out of its membrane, and they make a corona around it, or a crown around it, which is a derivation of its name. And viruses are simple protein with nucleic acids on the inside, and the nucleic acids are their gene. So they have genes comprised of the same nucleic acid molecules, whether it's RNA or DNA, that humans have. And the gene of the coronavirus is actually pretty large for the type of virus that it is. It has approximately 30,000 different 30,000 nucleotides. There are only 4 nucleotides, and they occur in all different sequences. But there are 30,000 reproductions of these nucleotides. And in the coronavirus, they make up approximately 10 genes. And each of these genes makes proteins. So there are obviously proteins for viral structure. So you have to have a spike, which is what we address with the vaccine. There's an envelope, there's matrix on the inside, there's nucleoprotein. All of these are structural elements to the virus. The virus also produces a number of enzymes, and those enzymes help package the genes inside the virus, and they help construct the virus when it enters the host cell in a human, so the infected cell in a human. So where you see these boxes like open reading frame 1, those are enzymes that are used for construction of the virus itself. And then you can see where the genes are for the spike or the nucleoprotein or the matrix across the whole gene, the whole genome of the virus. So the virus has 1 chromosome. We have 23 chromosomes. They have 1 chromosome, and their 10 genes are located on this 1 chromosome. Every time a virus divides, it produces a new genome, a new chromosome, and there are mutations. And with every division, mutations will occur. Sometimes those mutations are lethal. They occur in a spot where when that protein is produced, the protein is so malformed that it won't function, and that is a virus that is not propagated. Sometimes the mutations have no effect at all. They don't confer any benefit to the virus, and they're not lethal to the virus. And then other times the mutations do change the virus, and it improves the virus's ability to exist in its current environment. So you can understand that if you have a virus inside a human, and that human has been vaccinated, that virus is going to be killed. On the other hand, if a virus mutates to make it a little resistant to that vaccine, then that virus is going to persist. And the more it persists, the more it replicates. And the more it replicates, the more likely it is to produce another mutation, which will add a little more persistence to that virus in the presence of antibody induced by disease or by vaccination. This whole idea of mutation and persistence of viruses pertains really to all microorganisms. And you say, gee, how does this happen? How does it have a chance to really happen? But if you think, every plant and every animal has a nucleus full of genes. And every time that reproduces, we get mutations. So humans reproduce, once every 20 or 30 years, every generation, and each of our offspring have mutations. And some of those mutations, again, are important, some aren't important, and some are lethal. So there may be stillbirth because of the mutation. Every plant has mutations. Every bacteria has mutations, and bacteria replicate in hours. So if you think that humans have 10 times the number of bacteria in them and on them than they have cells in their body, every cubic inch of water, including the oceans, has billions and billions of bacteria in it. Every cubic inch of soil has billions and billions of bacteria in it. And every few hours when those things replicate, there are mutations in those genes. And to top all of that off, every bacteria, whether it's in water, soil, or in us, is infected with their own viruses. They're called phages. So bacteria have viruses also. And viruses go into a bacterial cell, and an hour later, they produce thousands of new viruses that go into additional bacterial cells. So the point I'm trying to get across is that there are a lot of genes out there, and there are a lot of opportunities for those genes to mutate. And we're just looking at one little opportunity here that the coronavirus has when it replicates inside of people who are sick with the virus. And one final thought is that you say, well, gee, what does a bacterial gene have to do with us? Or what does a viral gene have to do with us? It's now estimated that approximately 10 to 15% of our own chromosomes are actually viral genes. So not only do these replicate and mutate all over Earth, but they're swapped among organisms all over Earth. So if that's not enough to sober you up about genes and gene mutation, I don't think anything will.
So there are a couple of thoughts that I had along those lines that I want to make sure we just touch on. And I know we're running into our question and answer period. So one of the things when the virus first breaks out in China, it is unusual for a virus to mutate in a way that if it's leaping from animal to human, usually that's the mutation where it can actually then infect a human body and that person can get sick. The mutation required to then make it become airborne from human to human doesn't usually run concurrent with the mutation that allows it to go from animal to human. So Ebola is a perfect example. The human to human exposure is always through blood or whatever, that it's not, I can't breathe Ebola on you. And so one of the things with this particular virus was that it did both mutations, that it actually successfully mutated in a way that moved it from animals into humans and that it moved it from human to human through particulates. And then the second thing that they're looking at when they're talking about the mutations now is because we don't have herd immunity, this is a new thing in the human body, all of these people who are getting ill, not only is there just the risk to the human population because of the general illness, but it is that increased opportunity for mutations to take hold because there are millions of people carrying the virus right now. And that's one of the things that we're trying to get ahead of as well with the immunization program is to slow down the opportunity that the virus has to mutate. And I don't know if you want to add anything to that.
Well, no, that's excellent. Those are really very good points. We have 23andMe chromosome gene analysis for humans. They do that same type of analysis on viruses. And that is the way that they're able to track this virus. They're able to track it back, decades and centuries ago because they can track the mutations that have occurred. And the number of mutations will indicate how long ago this virus actually existed. So when we look at the virus in China, we look at the virus in the United States, we look at the virus in Europe, they're actually able to track the virus because of the mutations that occur. And among those mutations were mutations for increased communicability, primarily because people who are sick with these mutants have higher concentrations of virus in the respiratory tract. So when they dispel these viruses into the air, there are more viruses there. And that's one of the reasons that they have attributed to the increased communicability of this virus.
All right. So do you want to talk to this line? This is one of my favorites. in a horrifying way.
Please add to my remarks. This sort of reminds me that I have 2 grandchildren, Will and Annie, that look as innocent as these 2 kids do. These are 2 twin boys that one was vaccinated for smallpox and one was not. And you can see the effects of this vaccine on these kids. And if this isn't a perfect sales pitch for the value of vaccination, I'm really not sure what is. It's really quite a moving picture to see.
Well, and then that is in fact how they used it. It was actually used to market getting your kids immunized because they wanted to show the impact of protection by the immunization. And so this was a highly publicized photograph that went out at the point where smallpox vaccines were being rolled out.
Yeah, that's great. That's really very nice.
All right, and then lastly, wait, can I get to the last slide? There we go.
I just wanted everybody to know that, if you're still awake and really interested in this, which I hope you are, where there are 2 slide sets on the website for the History Center, and they contain more information than we were able to present tonight. So if you want some extra information or if you want references for the information that we presented, please visit the website and look these up.
All right, so we're going to go back to being able to see everybody. And for those of you, again, because there are a whole lot of human beings on this, you can click on the view and then you can go to speaker view and then you will be able to see who is speaking instead of looking at the multiple of the Brady Bunches on your screen.
They can send in questions, written questions through chat also, can't they? Or are we not handling those?
Yes, you can do both questions through that. And then we also have a compliment on your breadth of knowledge plus your handsomeness. What advice do you give to a person who does not want to get the COVID vaccine?
Well, my first advice would be get the vaccine. My second advice would be to talk to your primary care physician to see if you have reasons that the vaccine is contraindicated. And if not, to have a conversation about, why it would be important to get the vaccine. Unless there's a contraindication, I'm really in favor of people getting the vaccine. And this may take some conversation and getting comfortable with it and talking to peers, but talk to your doctor. It's sort of like the fear of flying. I mean, there are people that don't like to fly, but you can go through a gradual introduction to flying and become comfortable with it. I think the vaccine is safe and effective, and the sooner you can get it, not only does it benefit you, but it benefits everybody else.
All right, so the Greenwoods have raised their hand, but there was one quick clarification question. A contraindication is something that you have that means you shouldn't do something. It means that it is advised against. So if I have an upper respiratory infection, that is a contraindication for me getting a flu vaccine at the time because it could negatively impact me. I have to wait until I'm healthy. So that's what that's just a medical word that they use for meaning recommended against. All right, the Greenwoods. Please unmute yourself.
Yes, thank you.
There you go.
So I was on a Zoom call yesterday. I'm on the board of a nonprofit here in Philadelphia, and this was a new CEO who had been recruited to come from a museum in Wyoming to Philadelphia, and he made an interesting comment that the vast majority of their emergency responders, policemen, and I'm going to say hospital people, but I'm not sure that's the case. But a really significant number of people in Wyoming are anti-vaxxers, and he couldn't really provide an explanation. But he said that's a political statement, and I wonder how we deal with that.
Yeah, well,
in terms of herd immunity.
If I could answer that question, Mr. Greenwood, I'd,
I'm sure you can.
I wouldn't be retired sitting in my house. I think this is not just a problem in Wyoming, but this is a problem in different areas and around the country. And it's even, in Chicago and in New York. I have a microbiology friend of mine in New York who said that there were a significant number of people in their laboratory that elected not yet to get the vaccination. And the way they addressed that is that they brought in experts from the hospital, they're infectious disease clinicians, and they gave lectures on the vaccine and its safety and its effects and, the obligation of people. And they're slowly trying to convince these people. Now, in some hospitals for some vaccinations, you have to get the vaccine or you can't work there. Now, they haven't made that step here, and I don't think that they will. But, it's a slow process of education and people coming around to saying, gee, this is pretty good, we're going to use it.
All right, so we have a bunch of questions around the process of manufacturing. The first is, one person had heard that the size of space needed to cultivate the number of the volume of culture for this is the size of multiple football fields. Another person wants to know, is one vaccine going to be any better than the 2-stage one? And then the last one, which is the one that I don't have the answer to, but you do. Where do guinea pigs come from? Where do these first people who's like, yeah, you have no idea if this is going to work, I'm signing me up and I'm going to do maybe one of the first 10. Who are those folks? And then what is their long-term care should it not be effective?
Well, I wasn't part of the trial, so I don't know what the form was that I signed to see what type of care I would have if something went wrong. But I do know that all these are volunteers and that they had way more volunteers than they needed. They were able to recruit tens of thousands of people into these studies because at the other end of the vax spectrum, there are people who are really interested in getting these vaccines made effectively, and so they volunteer to do it. The newer vaccines don't take as much space to make as the older vaccines because we don't have to grow the virus in cell culture, the special way that we grow the virus, or grow the bacteria in broth or on plates. We're just taking a gene and we're replicating it in another microorganism. It's actually a pretty efficient way to do it. Now, it may take football fields worth of space because we're making so much vaccine. I've seen now that the companies that are producing the vaccine are now being reinforced by other companies that are not making the vaccine in order to increase production goals. Was there a third question in there?
Well, no, but I think you covered everything. Yeah. Go.
Vaccine versus the 2-shot vaccines.
Oh, yes.
Yeah. So I think that all the vaccines are excellent. And we look at these numbers, they might say 65% versus 95% or 80% versus 95%. They're not necessarily apples to apples because, for example, the single-shot vaccine, a big portion of those volunteers were in South Africa where they have more variants than they do in the United States. So if they actually do a trial where the vaccine hasn't mutated as much, it may look better than if it is an area where there's more of the mutants. So the experts have concluded that all of these vaccines work and that everybody should get a vaccine as quickly as possible, regardless of the vaccine. I'd be willing to bet that if a year from now, 9 months from now, 6 months from now, you wanted to get boosted by a different vaccine because you didn't like the one you got, that you'd be able to go ahead and do it. I think just starting now is most important.
Well, and so I'm going to hook one of the questions onto this, which is help us understand herd immunity. And there are 2 ways for the population to get herd immunity, one through immunization, the other through exposure. So can you talk a little about what that looks like in the population?
Yeah, herd immunity really refers to the percentage of people who've had real infection, plus the percentage of people that have been vaccinated. And when that percentage is high enough that it prevents the transmission of virus within the community and essentially stops the viral spread. And based on how easy that virus is to spread or how difficult it is to spread from person to person, those numbers for herd immunity are going to vary. So the influenza virus may have a different herd immunity percentage than the coronavirus might have. I think we've learned something interesting this winter. The masks and the social distancing have essentially eliminated influenza. There is very, very little influenza this winter compared to all other winters. And people feel that that's because of the steps we've taken to prevent the transmission of the coronavirus. Now, even with those steps, we have more transmission of coronavirus. We have ongoing transmission of coronavirus, yet we have essentially no transmission of influenza virus. So that tells me that influenza is less communicable than this coronavirus is. And if you were to translate that into what percentage of people would have to be immunized naturally or by vaccine before you stop the spread, it probably has to be higher for coronavirus than it does for influenza virus. And we don't know what that percentage yet is for coronavirus.
So one question, how effective is the COVID vaccine for people who are immunocompromised?
Well, this I don't know, and you'd have to talk to your physician about that. It would depend upon your immunocompromise and whether or not it's permanent, long-lasting, B cell, T cell. There's a lot of individual characteristics that go into being immunocompromised. Some of those will still allow you to produce antibody if immunized. Some of those will diminish the amount of antibody that you would use. And that's all a conversation you have to have with your clinician.
All right, and then somebody had asked a question about, okay, so we're all getting immunized, but when they release the version that is allowed to go into children's bodies, how confident are we that we know that that one is going to be safe and effective?
Well, I think you're going to be as confident as you are with the vaccine being released into adults. It is a rigorous hurdle for vaccines to get over new drug approval. And we now have vaccines that I think are down to age 18. Maybe the one-dose vaccine is down to 16, but we're obviously looking for a vaccine that goes down to 1 year old or so we can vaccinate people as soon as they no longer have their mother's protection. And they will do appropriate trials to make sure. Once again, it's a rigorous path, and we just have to listen to the scientists and the experts when they explain the results, to have the confirmation that they are safe.
And then another question, so can you get the vaccine if you're pregnant, particularly if you're early in your pregnancy?
Yeah, so again, I'm not the one to ask. You should ask your gynecologist, your obstetrician. But pregnant women were not included in the early vaccine trials. I know that some trials underway right now, they are including pregnant women. The Society of Obstetrics and Gynecology has come out and said that women can get it, but they have to talk to their obstetrician about getting the vaccine before they make that decision.
All right, and so there are a couple of questions that are tied around explaining the difference between the three current vaccines. So the Pfizer, the Moderna, and then the new Johnson & Johnson. Two are mRNA, and then Johnson & Johnson is a more traditional manufacturing process.
No, that's a new manufacturing process also.
Okay.
So the mRNA is pretty cool. The RNA is the gene of the coronavirus, and they take the portion of the gene that produces the spike, and they put that inside a little lipid droplet and they stick it into your arm. And lo and behold, that lipid droplet with that little gene in there is picked up by cells in your arm. And then the gene is actually, protein is made from that gene, just as the cell is making proteins from the genes in the your nucleus of that cell. So the proteins are incidentally made, they're excreted from the cell, and there they are for your body to react to because they're a foreign particle and you produce antibody. It's really remarkable. It's really a remarkable vaccine. The Johnson & Johnson vaccine is an adenovirus vector vaccine. So they take an animal adenovirus that is not pathogenic in humans, and in that virus, they put the mRNA, they put the gene for the spike protein. So when that adenovirus is stuck into your arm, the adenovirus genes are reproduced in your arm, and that gene for the spike protein is produced, it's secreted, and it's there as a foreign particle, and your body produces antibody to it. It's remarkable. Both of them are absolutely remarkable, and this is the first time that both of them have been used. They've been developed before, but they haven't been used in widespread vaccination program like this.
All right, and so what we're going to do one last question because I know we are running out of time. But one person asked, they didn't actually have the ability to identify influenza in 1918. So how do we know that that was what was spread and that it wasn't a form of coronavirus?
Yeah, so that's a really good question. And there are what we consider to be influenza pandemics in decades and centuries prior to 1918 that some people feel may have been one of these rogue coronaviruses that jumped into the human population. And over the years, it's become less pathogenic and is actually one of the coronaviruses that's out there now that just causes the common cold. But the 1980 influenza virus, we have been able to sequence the genes, reproduce all the genes in that influenza virus from mummified remains of people who died of influenza back in 1918. So we actually recreated the whole virus through molecular techniques, and presto came up with the H1N1 influenza virus that caused the pandemic. In addition, you can look at antibodies that older people have, and you can tie those antibodies to various epidemics that occurred throughout their lifetime. And so they put all this information together, and they have confirmed, even though we didn't know what influenza, it was a virus back then, we've confirmed that it truly was an influenza virus.
All right, although I know we've got some other questions, I do want to make sure that we can wrap this up. We promised everybody an hour, and I know that we've got some folks who are going to stay on and have a conversation following this. Thank you so much, Tom. I think people really enjoyed it. We got a lot of very good questions and a lot of very, very positive feedback about the way you delivered the information. Really helpful.
People who have questions that weren't answered want to email that question to you guys, and you share it with me. I'd be happy to send them an answer.
All right, I'm going to share my email on the chat. And also you can find us online at lbhistory.org. And then I will be happy to pass them on and get them to everybody.
Great. Thank you. Thanks, everybody.
Thank you so much.
Thank you.